Buon pomeriggio a tutti, questo è il quarto degli 8 seminari degli Spring Debates 2026 che sono organizzati ormai da 5 anni dal nostro dipartimento, il Dipartimento di Scienze Ecologiche e Biologiche dell'Università degli Studi della Tuscia. 4 dei seminari di quest'anno sono erogati in lingua inglese, quello di oggi è uno di questi, 4 sono erogati in lingua italiana, ogni 2 settimane c'è un seminario, arriveremo fino al 26 giugno di quest'anno, quindi poco prima dell'estate. Questa è un'occasione che noi riteniamo molto importante per un confronto scientifico tra docenti e ricercatori della nostra università e colleghi da università straniere e nazionali, ma anche e soprattutto con gli studenti, molti dei quali sono estremamente interessati soprattutto alla prima parte di questi seminari dove questo nuovo format ci permette di raccontare le storie personali e professionali dei ricercatori e dei docenti che ci raccontano come si diventa ricercatori e docenti e spesso, insomma, seppur aneddotiche, queste storie danno degli spunti importanti ai ragazzi più giovani. Passiamo in inglese. Today's seminar is given by the Associate Professor Jonna Kulmuni. Please forgive me if I pronunciate your name or surname incorrectly. I try my best. And I'm not going to present her much because actually she will provide all of the details later on. But just for everybody to know, she's an evolutionary ecologist. She works on genetics and evolutionary processes, including hybridization that she will talk about today, and about how genomes and biodiversity act in natural populations. Her title of a talk, this is what I got from a page. The title of a talk today is Hybridization and the Persistence of Biodiversity. So very timely topic and of interest for many. I thank everybody for being here and for people that will join us on the way of the seminar. Again, you will find the video on YouTube if you want to follow the first part. I take also a chance to thank our colleague, Professor Paolo Momigliano, who kindly contacted Jonna and invited her to participate to this seminar. This is really great. Thank you so much for being here. And I leave you the chair, and please everybody enjoy the seminar and take a chance to ask questions live when you can.
Thank you very much. Thank you for the invitation. I am sorry that I cannot be there in person, but I'm happy to be able to interact with you online. So I'm an associate professor at the University of Amsterdam currently, and at the same time, I'm still a group leader at the University of Helsinki. So I have this dual role for a little while still. But I was asked to tell a little bit about my personal journey in the beginning. So I will do that. So I started my PhD in 2007 and defended in 2013, which is actually quite a long period if you consider nowadays. PhDs are supposed to be 4 years in many countries. But I think I had the luxury of actually living and being in Finland. So I had 2 kids during my PhD. I was able to take 10 months maternity leave with both kids. And on top of that, I did a second master's degree while I was doing my PhD. And this was a master's degree in science communication, which has actually come very handy in the scientific career as well. But I think essentially like being in Finland gave me this freedom to have these other aspects of my life during my PhD. My PhD was on ants. Half of it was looking at questions related to speciation and hybridization. Half of it was evolution of gene families, so very molecular. And after my PhD, I really thought long and hard, what is the direction I want to take next? And at this time in our field, there were sort of technology had developed so that it was able to sort of study natural selection in action from genomes. So you could sequence genomes at the beginning of the experiment and follow the rise of mutations over time. And this was mainly done in microbes. And I was super enthusiastic about this. And I was thinking that I will drop the ants and I will move to microbial research. But then I had a chat with my office mate, and she said that, you are in a luxurious position that you are working on a model system, the wood ants, that if you don't continue this, no one else will. because my supervisor had retired. There wasn't anyone really continuing to address the questions that were left after my PhD. So I decided I stick with the ants, but I put them in the general context of speciation. And the reasoning for that was sort of twofold. People who study social insects, they are interested in the evolution of sociality, not really speciation. And then if I looked at the speciation as a field, there were many different model systems, but no one was really using ants to address general speciation questions. So I decided that I will develop my own research questions. I applied funding and I went to the University of Sheffield to work with Roger Butlin, who is one of the best known speciation biologists in the world, I guess. And we were there, so we moved there when my kids were 1 and 3 years old. I made a deal with my husband that if we go to Sheffield, after 2 years, he can decide whether we stay in UK or whether we go back to Finland. And he decided that we go back to Finland. I wanted to stay, but he thought that job opportunities are better for him in Finland. So we went back, and I was again a little bit in this situation of, thinking like, what is my strategy? And I decided to put all my efforts in really continuing to develop my own research program. I applied various funding. I was successful in some of them. And in this, on green, you can see all the funding I've applied. And it really takes, you see the successful funding, but it really takes a lot of applications to get one successful funding. So I was in Finland for several years and was quite successful with the funding. And in Finland, you can build your own research group without having a permanent position, any faculty position. And I had applied all the jobs that were open in Finland for any university in Finland, and I didn't get any jobs. So around 2022, I said to my husband that, look, if I want to continue in science, I think I have to apply jobs outside of Finland. And together we made a strategy of the countries that we could imagine ourselves living in. And I applied my first job that was in the Netherlands, and I got it. So in 2023, we moved to the Netherlands with the whole family. My daughter wished that we could have moved to California, but I guess you can see that Netherlands is sort of, it's the west coast of Europe. Here in Amsterdam, I've gained a very good community. I really enjoy professionally working here. And I have gained also a new model system that I'm super excited about, because now with that system, I can actually do what I wanted to do with the microbial evolution. And in this 10 years time, I have submitted at least 36 grants. I've gotten 11. So it takes, many tries. And then to sort of briefly summarize, if I think about my guiding principles, well, maybe you can guess the first one. So family comes first to me. It's important to take the entire family into the decision making. The second one is do not work with assholes. I work with people who I can trust, have the same values as me. And actually, I've put a lot of effort in building a network of international collaborations. And I really enjoy that network and benefit from that. And lastly, do fun things. So follow your, the questions that you are interested in. That's something that I've been doing, even though it wouldn't necessarily be the hot topic where everyone is working on, but something that you are interested in will motivate at least me further. So this was the bit about my professional journey, and maybe just briefly to ask if someone has questions, I'm happy to address those now about the personal journey.
I love the guiding principles.
Okay, if no one has any questions at the moment, yeah, you can ask also at the end. So perhaps I will then move on to the sort of more scientific part of the presentation, which I titled Cost and Benefits of Hybridization for the Persistence of Biodiversity. And I will be combining approaches that we've used with the wood ants, which is a system I've studied for over 20 years now, the two-spotted spider mite, which is a new system for me, and modeling and simulations that have actually become quite an important part because there is this feedback loop with models and simulations and experimental work. So let me put this one. So hybridization is the interbreeding between genetically distinct populations producing offspring of mixed ancestry. It was once thought to be rare, and because of the deleterious consequences it can have, it was considered an evolutionary dead end. However, with the possibility to sequence genomes from non-model organisms, it has become apparent that traces of past hybridization and gene flow characterize the history of many taxa, including our own species. But what were the initial conditions that led to these outcomes? In many cases, we don't know that. Rates of hybridization are further expected to increase because of human action and ongoing climate change lead to the movement of species and populations, providing opportunities for hybridization. But are these increased rates of hybridization a bad thing? Sometimes they are, because hybridization can lead to the breakdown. Wait a second, I will just move you. Breakdown, like in the case of plant hybrid necrosis, like in the picture, or in the worst case scenario, hybridization could result in extinction of populations or entire species. But then on the other hand, hybridization may also provide benefits by boosting speciation and adaptation. Take, for instance, the wood ants. We have shown that despite costs, hybrid populations persist in nature. Wood ant hybrids also have higher genetic diversity and heat tolerance compared to the parental species, and all of this could benefit the hybrids as climate warming continues. But at the moment, we don't know when the benefits of hybridization outweigh the costs. We cannot predict this in most cases because studies up to date, they are restricted on narrow snapshots of time or space. There are few model organisms, for example, like the yeast, where we have bridged this gap, but we lack essentially the ability to generalize outcomes of hybridization across sexually reproducing eukaryotic taxa. And upon hybridization, what happens is that two gene pools are combined, which results in increased genetic variation and new allelic combinations. Both of these could boost rapid adaptation. However, some of these new allelic combinations may not work well together and result in hybrid incompatibilities and breakdown. Like in the case of a classical Dobzhansky-Müller incompatibility, you have an ancestral population. It divides into two populations. In one of the populations, this red allele arises and fixes. In another population, blue allele arises and fixes. And if these alleles are brought together in hybrids, they have never co-evolved, and they might result in a deleterious fitness outcome. But importantly, during hybridization, both deleterious, beneficial, and neutral allelic combinations are generated simultaneously, And it's sort of this combined effect of all of them that make up the hybrid fitness. And these fitness benefits and cost of hybridization, they can vary across the genome, over generations, and across different environments. And I think it's these three sort of axes that we need to be able to understand to predict the scenarios when the benefits of hybridization outweigh the costs. Speciation studies have utilized hybrids to map barriers for gene flow and loci that underlie reproductive isolation and hybrid breakdown. And this means that hybrid fitness has been measured mostly in the parental environment, because this has been interesting for speciation. However, at the time of environmental change, understanding hybrid fitness in novel environments becomes important. For example, in hot environments or environments that experience a lot of variation. And a hypothesis that we've been working on in my group has been that actually it could be that the benefits of hybridization outweigh the costs, especially in novel environments. And to test this idea, a few years back, I visited Sally Otto's lab. So she's a world-leading biomathematician, and I went to her like, look, you know, I'm actually quite bad in mathematics. I would like to learn some simulations. And together with her and her brilliant, then a master's student, Bryn Wiley, we performed some simulations. So to look at hybrid fitness in novel environments, we used Fisher's geometric model. So this is a model where you have a fitness landscape, like the one in the picture, and mutations take you steps along this landscape. So mutations have additive effects. So they can take you on top of that high fitness peak, but they might have non-additive effect for fitness. So imagine you are on top of that high fitness peak. If a mutation takes you a little bit outside, your fitness drops tremendously. So in a way, this is a model that naturally incorporates epistasis, which is the essence of hybrid incompatibilities. This is the model that we simulated. So we started with two parental populations. They were evolving without gene flow. So they evolved in allopatry. At the beginning of the simulation, we put them in a new environment, which is this blue environment. We let the parental populations adapt to that environment. And then at generation 1500, we generate a hybrid population from those parents. And we changed the environment again. So we put everyone into a new environment and look who adapts fastest. Is it the hybrids or is it the parental populations? And in these simulations, we varied the parental histories so that sometimes the parents were adapting to the same environment, or maybe they were adapting to completely different environments. So we varied the parental history. And we also did these simulations in both diploids and haplodiploid organisms, because actually both the wood ants and the spider mites are haplodiploid organisms. And I'll orient you to this first figure of the results. So on the x-axis, you have generations starting from the generation 1500, where hybrids were made and everyone was put into a new environment. You have the fitness on the y-axis. And in each panel, we have a sort of different scenario of parental adaptation. But essentially, from the beginning, in always these figures, you have hybrids on lighter colors and parents on darker colors. And what you can see is that the hybrid fitness increases faster than parental fitness in this new environment. And this result actually applies to nearly all simulated scenarios. So you can see that in almost all of these figures, the lighter lines are higher than the darker lines. So on top row, you have the diploids, bottom row, you have the haplodiploids. The general same principles apply to both. The only scenario where hybrids don't adapt faster is this one, the C one, and that's where the new environment is actually quite close to the environment where parents had already adapted to. So that's a scenario where hybrids don't have an advantage. But what these simulations also revealed to me was that hybrid breakdown actually has a relatively minor effect compared to the effect in the reduction of fitness that's caused by this new environment. And furthermore, these results remain if we do the simulations with incompatibilities that don't depend on the environment. So in a way, in this modeling framework we used, the incompatibilities, this basis of breakdown is environment dependent. But on top of that, we introduce incompatibilities that are always bad. And despite these, hybrids adapt faster. So I thought that this was quite an interesting result. Okay, hybrids actually have an advantage in new environments. And the question I had, well, what have we missed? Do these simulations apply to real populations? And at that time, I arrived to the University of Amsterdam and found the two-spotted spider mite system. And this is actually an ideal model for experimental hybridization because it has a short generation time, so it can be used to study outcomes of hybridization over tens of generations with high replication. We can estimate individual fitness, and these herbivores can be grown on different host plants, which makes it easy to study fitness consequences of hybridization in different environments. Finally, despite their tiny size, it is possible to sequence the genome of an individual mite. And by looking at the genome, we can actually then track in our experiments, which genotypes, which hybrid genotypes are being favored and result in the higher adaptive potential of hybrids. So we have done some sort of pilot experiments to test if the benefits of hybridization outweigh the costs in novel environments. And this is work that was started by Jonna as a master's student when I also started in Amsterdam, and it has been continued by Marit, who is a PhD student with me working on the spider mite system. So in these pilot experiments, we crossed two spider mite lines. One originates from London, Canada, the other one from Zandpoort, Netherlands. And both of these parental populations, they've been kept in the lab on bean. So they are herbivorous insects. They need a host plant. They eat that plant. And both of these parental populations had adapted to bean. But we were interested in hybrid and parental fitness on novel host plants. So we used quinoa and maize as novel host plants. And then this is the cool thing about the spider mites. So this is the fact that you can measure individual fitness of mites on these tiny leaf discs that you see in the picture. And you can have mite fitness on parental host, you can try it on the novel host, and then count the number of eggs that were laid, the number of eggs that hatch, and the number of individuals that survive into adulthood. And this is exactly what we did. So in the first pilot experiment, we made hybrids. We had hybrids and the parents on the parental host bean, and we measured their fitness in the first 3 generations. And what you can see is that parent has significantly higher fitness than hybrids on the parental host. So this is a clear signature of hybrid breakdown. Something is going wrong in these hybrids. However, if we have a look at the fitness of parents and hybrids on this novel host, actually there's no difference between the parents and hybrids. Both of them have equally low fitness. After this first pilot experiment, due to resources, we weren't able to grow and keep these on the different host plants and follow the fitness in the lab. So both the parents and the hybrids, they were transformed into bean, and they were kept on bean plants for the next 40 generations before performing the second pilot. And in the second pilot experiment, now the axes are a little bit different. So this is 1 generation in days, and on the y-axis you have the percentage of eggs hatched and survived. So again, a proxy of fitness. But what you can see is that on the parental host, actually now hybrids don't suffer from this breakdown anymore. So they've overcome hybrid breakdown and have a similar fitness to their parents. However, if we look at hybrids and parents on a novel host plants, which this time it was maize because it was available in the lab, we see a tiny difference where hybrids actually do slightly better on this novel host plant compared to the parents. So now, based on these pilot experiments, we've designed and are doing the experiment again, this time actually measuring fitness every generation so that we can bridge that gap where we think that hybrids overcame the incompatibilities. So what we gather from these pilot experiments is that the costs and benefits of hybridization, they vary across generations and environments in the spider mites. But in the future, I think the questions what we want to address is, is this overcoming of hybrid incompatibilities, is that repeatable? Do they consistently get rid of these incompatibilities? And what's the time frame for that? How many generations does it take until hybrids can recover from these incompatibilities? And then if we look at the benefits of hybridization, how fast does this happen? Is this also repeatable? And what determines the benefits of hybridization? And interestingly enough, we know something about these questions in a system that you cannot control, you cannot do experiments with, but we are actually just relying on what we find in the nature. And those are the mound-building wood ants. So I've studied these mound-building wood ants since the beginning of my master's thesis. And in the picture, you see one of our experimental nests. So it's behind here. And this is a nest where I've been going since my master's studies, where we go with my group still. And these are hybrids, and we've sort of focused mostly on hybridization between 2 species, Formica aquilonia, which is a northern species and cold adapted, and Formica polyctena, which is a southern species and warm adapted. And their overlapping range is on the light green. It doesn't necessarily mean that there are hybrids there, but that's where their ranges of the parental species should overlap. And especially in southern Finland, we have studied and monitored hybrid populations for 20 years now. What we know of the parental species, they are both supercolonial and they have many queens within their nest. So this means that a population consists of a network of nests that are interconnected, and each nest can have hundreds of reproductive queens. We have looked at the history of these 2 species by demographic modeling and shown that they have diverged with gene flow in the past 500,000 years. We also know that the parental species are divergently adapted to climate. So Formica aquilonia indeed does better in cold tolerance assays. Formica polyctena does better in heat shock assays. And we've sort of, through our studies, accumulated evidence that hybridization results both in costs and benefits in the wood ants. So we have shown hybrid breakdown, especially in the males, but then again, hybrid vigor in the hybrid females. If we look at the hybrids and compare them to locally dominant parental species, Formica aquilonia, hybrids have a higher heat tolerance, and they occur in habitats like microclimates that are warmer compared to the cold-adapted parent. And our recent studies show that hybrids are actually rapidly expanding their range northwards in Finland, and this coincides with climate warming. So it really looks like that hybrids may have some advantages in warming climate. But what I wanted to focus on today was actually more genomic information, genomic data. And this starts from a study. So I'll show you unpublished data, but it sort of starts from a study that we published a few years ago, where we showed that actually hybrid genomes in 3 independent populations evolve in a correlated manner, evolve in a predictable way. So in this study, we had parental species, both from Finland and outside of Finland. And we had individuals from 3 hybrid populations that were located here and quite close to each other. We sequenced the whole genomes of hybrids and parents, and what we can see, so this is a PCA based on the whole genome data, is that the hybrid populations situate in between of the parental species, so northern parental species, southern parental species, and furthermore, the hybrid populations cluster within a population. So they are genetically differentiated from each other. So one population is here, the other one is the pink one, and the second population has both red and blue dots in it. That's because these are 2 hybrid lineages that originated from the same hybridization event, coexist in the same population, but don't really interbreed with each other. But when you compare the genomes of these hybrids, so let me orient you with this figure. So you have the part of the chromosome laid on the x-axis and the 3 different hybrid populations on the y-axis. And then you can imagine that you see the relative proportions of parental ancestry indicated with the 2 colors. So if there is yellow, it means that the populations have ancestry from Formica polyctena. If there's green, it means that the hybrid populations have ancestry from Formica aquilonia in this part of the genome. And our demographic modeling and genetic data shows that these are probably hybrid populations that originated independently from separate hybridization events, and they have since evolved without gene flow from other populations. So they are sort of experimental replicates in nature. But still, you see this repeated pattern in their genomes. So, for example, this part of the genome, most populations or all of these populations, they have fixed mostly ancestry from the northern parental species. Whereas, for example, in this part of the genome, there is an area of the genome where they have fixed mostly southern parental species ancestry. And this was quite surprising to us. We sort of thought about different drivers of this sort of sorting of parental ancestry. And we got support for one reason why the populations are evolving in a correlated manner is that they are masking or getting rid of deleterious alleles from the parental species that has a low effective population size. So that is Formica polyctena, which is then expected to have fixed slightly deleterious mutations. And once you hybridize, you can get rid of these mutations because you can choose and select for the other parental ancestry. We also saw that the hybrid populations were fixing the parental haplotype that had a signature of recent positive selection. So if there was a sort of genomic region where Formica aquilonia had experienced a selective sweep in the recent history, then hybrid populations were more likely to fix this haplotype. So it sort of suggests that the hybrids can sort of choose from the best combinations of haplotypes. And that's why they are partly evolving in a repeated manner. Okay, so this was in 3 hybrid populations. Now our goal is to expand. So if we expand the sampling and we take more hybrid populations that occur in different types of environments, can we still see that they are evolving in a correlated manner? And can we actually disentangle the role of extrinsic selective pressures and intrinsic selective pressures that might drive this correlated evolution of hybrid genomes? So this is work that's ongoing. These are unpublished results, and this is a work that's led by a postdoc, Noora Poikela, in my group in Finland. And in this study, we now have expanded to 18 different hybrid populations. We have most of our populations from Finland, but now from a larger geographical area, and then few populations from Central Europe. And we sequenced about 10 individuals per hybrid population. Again, what you can see is that the hybrid populations are genetically distinct. So here again, you have the parental species, southern parental species on yellow, northern parental species on green, and hybrid populations with different colors. And what you can see is that the hybrids sort of cluster within their own color, suggesting they cluster within their own population. Another way to visualize this is the admixture plots. So here you can imagine that each individual genome is represented by a vertical line, yeah, and the composition of that genome is represented with a color. If it has the same color, they are genetically similar. So if we divide the genomic data into 2 clusters, which would be the parental species, we see the parent 1 here and the southern parent here at this other end. Whereas the hybrids, they have varying proportions of ancestry. If we divide the data into more genetic clusters, you can see that again, the individuals group by their population. This is the one exception, which is again, this population that has 2 genetic lineages, hybrid lineages. A further representation of the same data is here, where again, you see that individuals cluster by population. So we think that these populations, at least partly, they have evolved independently. So they are distinct. Another question that we had is that, do these hybrids represent different ages? And the reasoning here is that when you hybridize, you have 1 full chromosome from the yellow parent, the other full chromosome from the green parent, and if they interbreed with each other, with other hybrids in the same population, over time, these parental haplotypes are broken down. The smaller the haplotype, the older the hybrid population. So we looked at the length of these parental haplotype blocks, and we indeed see a distribution of different ages. So we have some populations that are very young, that have very few large that have very few small haplotypes. And then again, we have other populations that are clearly of different age. So what we think is that the hybrid populations have evolved independently, and they represent at least partly different ages. And the independent evolution is important because if we want to ask questions about repeatability of evolution, we need to have populations that are independently evolving. Okay, so then first look at the possible repeatability of evolution in these hybrids. So again, let me orient this figure. So this is a new method of genome polarization called DM. And essentially, it is reference-free, so you don't need to say that this is species 1, this is species 2, but you give the data and the program, the software, finds the loci that mostly clearly distinct groups in your data. And in this circular plot, now we have the entire ant genome represented in a circle. You have here on the outer side, you have the northern parental species. I'm sorry, the colors are flipped. And here you have the southern parental species. And in between these are all the hybrid individuals. And by first glance, it does look like there are regions of the genome where all the hybrid populations tend to fix same parent ancestry. You can look at the data in a different way. So this is similar to what I showed you before, a different way of looking at the parental ancestry in hybrids. And again, you can find these regions of the genome that look like, you know, all the hybrid populations have fixed certain parental ancestry. So it seems that these hybrid populations are evolving in a repeatable way. The next step that we need to do is to actually quantify that. We've now looked at the extrinsic selective pressures that might dictate the evolution of these hybrids, and one clear is climate. So we know that, you know, there are differences in climate between the parental species. So we took bioclimatic variables from the locations of the hybrid populations and made a PCA out of these bioclimatic variables. And this is how the hybrid populations situate in this sort of climate space. So PC1 is sort of axis of winter temperature, temperature variation, seasonality, and higher precipitation. Whereas the PC2 axis, it's an axis of cooler summers and lower precipitation. And if we now ask, can we find genomic regions that are sort of linked, underlie this climate adaptation in a sort of traditional GWAS manner, we do find many genomic regions throughout the genome that are potentially underlying climate adaptation. And the next steps here is to then look at those regions where we think climate is driving the evolution of those regions, look at those and see whether we find that there's sort of predictable parental haplotype combinations in certain climates. I will speed up a little bit. Last part was we've looked at intrinsic selective pressures. So we've mapped incompatibilities, the transcumular incompatibilities throughout the genome in another study. And they are essentially incompatibilities occur throughout the ant genome. It's a network of incompatibilities. And what we now want to do is to look at whether these incompatibilities are driving correlated sorting across these hybrid populations. So perhaps if the populations are sort of purging, getting rid of the incompatibilities, these regions could sort fastest in a predictable way. And tentatively, our preliminary analysis suggests that that is the case. So if we sort of put together all the different systems, so in the wood ants, we find that evolution takes sort of predictable and repeatable paths in hybrid populations. And my hope is that we can sort of disentangle the genomic signatures of benefits and costs with the wood ant system. In the spider mite system, we can then actually look at evolution in real time and quantify the cost and benefits of hybridization and how they depend on the generation and the environment. And sort of this feedback loop with the simulations helps us to understand, well, generate predictions for our experimental studies and vice versa. And how this all links to biodiversity, this is my last slide, is that understanding these costs and benefits of hybridization, it's not only interesting for speciation, but it has implications for the persistence of biodiversity in this rapidly changing world. So if we find that hybrids are better equipped to adapt to climate change and other anthropogenic threats, then they can be actually important for the persistence of biodiversity. So if we can predict the systems and scenarios where benefits of hybridization outweigh the costs, then we should actually include hybrids into our conservation planning, or maybe in some scenarios, they should be given a conservation priority. Thank you very much. And I'm happy to take any questions.
Thank you very much. What a wonderful seminar. I heard that your graphics were outstanding, but seeing them, like, I understand where the comment was coming from. Yeah,
Thank you. Thank you.
Somebody that doesn't know much about genomics, it's very easy to follow you up. Thank you so much for this presentation.